In this lecture, we finally get to protein folding and sequence design. So we're going to be modeling proteins in various different ways and then focusing on one or two ways to help determine the most probable sequence. So the first thing we're going to start off with in part one is modeling proteins through a helix coil or Coil-Helix transition, and then take a look at some of the energetics. Then in part two, which is chapter 7, we're going to look at the toy model of protein sequences, which is really fun to work with and very qualitative and visual. Okay, so where do we start? Let's quickly go through the main points from the previous lecture. We talked about protein structure, so understand the different types of proteins and their functions. We also talked about peptide bonds and alpha carbon bond angles. Those are important too because that creates the polypeptide sequence that we're going to look at for this lecture for part 1 and 2. So in the previous lecture, we took a look at how a sequence was formed with each of the amino acids being connected together through peptide bonds. Each of those alpha carbon bond angles had 2 degrees of freedom as a model, whether they can twist left or right with respect to each other. And then now we're going to take a look at protein folding. So once they start to fold, we're looking at secondary structures. So that would be alpha helices as well as beta sheets. From there, we're going to be talking about alpha helix to coil or coil to helix transitions and present a 2-state model for the alpha helix. So if you think about it, when you have a sequence that's a linear sequence and you want to turn it into a helical structure, there has to be an initiation step. So that means from a helix or coil form, we want to be able to transition between back and forth between each of them, whether it's uncoiling or coiled together. Then we're going to go through sequence predictions in part 2. So this is now something called protein-like sequence design. If we give you a set of amino acids, what is the most likely shape that it's going to form, and what is the most likely compact structure with the minimal amount of unfavorable energy. So a lot of these discussions and a lot of these optimizations have to do with energetics, and we're going to be talking about that at the end of each of those parts for each of the models. Okay, so let's now go straight into protein folding. We're going to first talk about what forces help proteins fold and stabilize them. Then we're going to look at what kind of secondary structures there are and what they form. So that would be the alpha helix and the beta sheet. And then we're going to take a look at higher level structures such as tertiary structures. So we're first going to take a look at why proteins fold, and this is a very important question to be able to answer. Primarily, they fold because of weak interactions. So we have to ask ourselves, what kind of interactions are there? In this image, we have a central protein, and basically what's happening here, so this is our central protein, we have electrostatic interactions or attractions taking place to connect these two sides together so that there is some promotion and encouragement of that sort of folding to take place. Over here, we have a hydrogen bond that takes place. These ones we've studied a lot before, so I won't go into detail. And also they provide weak interactions for two sides of the protein sequence to connect with each other. And then over here at the bottom, we have what's called van der Waals attractions. So these attractions are small, very weak attractions between close distance atoms and molecules. So it's almost like a cluster. So it's a combination of all of these weak interactions that create strong bonding arrangements for each protein. So it's not one by itself, usually it's a combination. Now let's introduce something else called hydrophobic forces. So we haven't really talked about this yet, but these are forces that help proteins fold into compact conformations. So by its name, we can probably assume it has something to do with hydrophilic or hydrophobic substances, and that's exactly what it is. So polar amino acids are going to want to be together, and non-polar amino acids are going to want to be together. So with our previous analysis into polar and nonpolar interactions, water is going to want to be around polar molecules because of higher entropy, and water will also want to group nonpolar molecules together to minimize its individual interaction with them. So based on that, it means that when you have nonpolar side chains, they will want to be buried inside a protein conformation to shield it from water because all proteins are in some sort of fluid. So the nonpolar side chains will want to be on the inside to form almost a hydrophobic core. And then on the exterior of the protein, there will be hydrophilic side chains that prefer interactions with polar molecules or with water. So just keep that in mind. This is a good picture to represent that. You have a protein that has been folded up, and you can see all of these green circles. These are the nonpolar side chains that have been packed into a hydrophobic core, and then you have all of these external hydrophilic or polar side chains that are facing outwards towards the water. So now that we've folded everything into a certain shape because of hydrophobic interactions, we still have to keep them in that form no matter what. So when your protein has folded into its desired shape, or its most probable shape, it's hydrogen bonds that help keep it together. So these occur between polypeptide backbones and or side chains, depending on their electronegativity, to stabilize a protein's folded conformation. So we've already covered how proteins can fold and how they stay folded. Now the next question would be, how can we guarantee proteins get folded properly? So there's something really cool called a chaperone protein. These assist in protein folding. So these chaperones, the namesake is very fitting, they basically help make sure an isolated polypeptide is folded correctly and they don't aggregate into non-functional structures. So looking at the diagram here, we can go through step by step. You have some newly synthesized and partially folded proteins, and it enters the a chaperone protein and basically get a clean environment to fold properly versus accidentally aggregating, like grouping together outside in the environment, or folding incorrectly. So it actually goes into one of these chaperone proteins to get a clean, stable environment to fold the way it should. After it's done, the chaperone protein is going to release that folded protein out so that it can do its function. The other cool thing about chaperone proteins is that a lot of them are heat shock proteins. So it's very important to understand why. Number one, why are heat shock proteins even created? Because of stress. So high temperatures, UV radiation, etc. Anything that can cause the denaturation of or misfolding of a protein, that is due to stress. So the connection to chaperone proteins is that chaperone proteins are there to guide protein folding and fix mistakes. So as stress increases, more misfolding and more issues or more aggregation will take place. So as a result, chaperone proteins are more likely to be required in stressful situations. And because of that, the majority of chaperone proteins are heat shock proteins, and they can exist in high stress environments to guarantee the proper folding of proteins. So moving on to what happens after a protein folds, it goes into what we call a secondary structure. So you've probably learned about this in previous biology courses or even in high school, but you've seen alpha helix and beta sheets as secondary structures. These basically result from hydrogen bonds that form between the amino and carboxyl terminus groups in the polypeptide backbone. On the left, you can see an alpha helix, while on the right, you can see a beta sheet. So we are going to analyze each of these separately. starting with the alpha helix. So looking at the alpha helix, we have hydrogen bonds between the N and the H. So you can see over here, the red lines, or the red dotted dashed lines refer to the hydrogen bonds. So that's between the NH and the CO groups in the polypeptide backbone. We can represent this as a helical structure. So we can simplify it to something like this, with each turn being 0.54 nanometers. So just another look at the hydrogen bonding arrangement. We can see that they occur in a stepwise fashion. So the first one is going to be hydrogen bonded to, let's say this is position 1, 2, 3, 4, 5. So 1 to 5, and then 2 to 6. So because of this patterned hydrogen bonding, it forms an alpha helical structure. But the other thing to note is when you turn something, it can be left-handed or right-handed. So the alpha helix, we have to understand there are two different ways of forming an alpha helix. It can be a right-handed alpha helix or a left-handed alpha helix. So if we know both can exist, the next question is which one is more common, or are they both equally likely? And remember, in the previous lecture, we talked about the Ramachandran plot. That's where that comes in handy. So in terms of what this plot does, it tells us what kind of bond angles are the most common. So if the bond angles are phi equals to 60 and psi equals to 60 right here, it seems like a left-handed helix can occur. Oh, sorry. Here, 60 and 60, plus 60 and plus 60. A left-handed helix can occur, but you can see it doesn't take up a lot of space, so it's probably very rare. Now, if we take a look at where the right-handed helix is, then you have minus 60 and minus 60 bond angles. And so this whole region is much darker. There's a larger, darker region, and that means that it's more stable and more probable to have a right-handed alpha helix. So based on this diagram, you can see the right-handed alpha helix is more common than the left-handed alpha helix because of preferred bond angles. There is one left. There is a giant blob on the top left. Because we are talking about secondary structures here for protein sequences, most likely this is going to be beta sheets, which we haven't covered yet. So before we talk about beta sheets, let's first wrap up what alpha helices and what alpha helical structures can do. So first, an alpha helix can cross a lipid bilayer. When we ask ourselves why, it's because it has hydrophobic side chains, which can insert into the hydrophobic core of the lipid bilayer. So remember, there's a difference between proteins folding completely in a 3D structure so that they have a hydrophobic core and the conformation of an alpha helix. So there's more steps for a protein to fully fold until it gets to its final state. The alpha helix is just one of those states or a part of a larger protein. So in terms of the alpha helix itself, because of the way it's coiled and the way it turns, most of the hydrophobic side chains are going to be on the exterior and thus allow the alpha helix to pass through the lipid membrane a little bit easier. So the other thing to note is that helices can intertwine with one another to create a coiled coil. So you can have two alpha helices, for example, and they can coil together and they create a much tougher strand or a more rigid strand. So this effectively minimizes the exposure of hydrophobic residues because you now have a partial hydrophobic core within each of these green areas. Okay, so the next secondary structure we want to talk about is the beta sheet. So once again, it's formed due to hydrogen bonds, but in a different location. It's arranged differently between the amino and carboxyl groups of the polypeptide backbone. Instead of forming a helical structure, it forms a sheet. So it kind of goes back and forth like this. So you can see this one, it starts here, and then another strand here and another strand, and then they have hydrogen bonds connecting each strand to each other. So your beta strands will now be the last blob that we saw in the Ramachandran plot. It tells us that the beta strands will be a much more favorable and stable conformation as compared to the alpha helix, whether it's left-handed or right-handed. But just like alpha helices have left-handed and right-handed versions, beta sheets also have different directions. And when you have sheets, you can probably call them parallel or anti-parallel. So the easiest one to look at first is the anti-parallel one, where you have a polypeptide chain going in this direction, then wrapping around in the opposite direction, and then going in this direction. So therefore, you're going to have hydrogen bonds between each of the neighboring strands that are kind of looping around. The other option for beta sheets is for it to be parallel, and this one's a little bit more complicated because you have to go all the way here in this one direction, loop all the way back, restart the chain in this direction, loop all the way back, and do it again over and over again. And you can see that the hydrogen bonding is going to be in adjacent regions between each sheet. So they occur between what we call alternative amino acid side chains instead of adjacent amino acid side chains. So finally, we've learned about alpha helices and beta sheets. We can combine them to create protein domains. These domains can come together to form larger protein molecules, and this final form is called a tertiary structure and usually has different functional domains that contain alpha helices, beta sheets, or eventually both. So now we can move on to modeling all of these. There are going to be several models that we present, some that are visual, some that are mathematical. So the next few sections will be quite long. First off, we're going to start with the two-state model of the alpha helix. We want to explain something called a coil-to-helix transition, and then use that knowledge to be able to predict the hydrogen bonding order of an alpha helix. At the end of this, we'll present a sample problem for hydrogen bonding sequences. Okay, so on the left side, I have a right-handed coil, just something that I drew. From before, we note that a right-handed coil was more favorable than the left-handed coil, so we might as well use that. And it was stabilized by hydrogen bonds. So why don't we take a look at the individual subunits that make up the entire helix and assume that they're in unity, because it's just a lot easier, and assume that they're all homogeneous. So the one thing we want to change is whether they're in a helix or coil state, but we have to define these first. When looking at a homogeneous polypeptide chain, we note that there can only be two forms of each monomer. So either the monomer is in a coil state, where the bond angles are free to rotate, so something like this, or the monomer is in a helix state, like this, which means that the bond angles are frozen due to the presence of hydrogen bonding, forcing it into a lower energy state. So when a monomer changes from coil to helix, or helix to coil, we call this a Coil-Helix transition. And of course, we call this a two-state model of an alpha helix. And this allows us to make various predictions about which conformations are possible or not. So starting from coil form, so disordered form, if we want to introduce some order to the system and create a helix, we're going to have to introduce some hydrogen bonding between different subunits within this coil. So it's starting from there that we can do all this modeling. So how do we do this? Let's first allow each monomer, which we're going to use blue circles for. We're going to allow each of these monomers to either be a helix state or a coil state. So only one of two states. So this is our monomer N. and we designate these as H or C, H for helix and C for coil. You'll see in the diagram here that there's a polypeptide chain, and we represent each amino acid or monomer as a blue circle labeled in the order of the chain. So starting at 1, this is the bottom, we have 1, 2, 3, 4, 5, and presumably 6, 7, 8, 9, 10 going along this way. There's also a green hydrogen bond. So this question or this problem already tells me where the hydrogen bond exists. It's between 1 and 5. So I can draw a hydrogen bond between 1 and 5 over here. So we can represent this in sequence form, where we draw one line to connect monomers 1 to 5. That's called sequence form. And then this is called the graphical form. And the overall conformation is called 1H. So that's because there's one hydrogen bond, and it's the first monomer that is in the hydrogen bond pair that's counted. So essentially, if I have this sequence here, 1, 2, 3, 4, 5, it's the first monomer that starts the hydrogen bond that's counted as an H for helix. So therefore, I have H, and then coil, 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 on until 10. So we say the first monomer is in the H state because the hydrogen bond promotes the helix transition, and the others are in the coil state. So it's number 1 that's in the helix state, creating the hydrogen bond that's pulling 5 towards it. So with our first model, we made the first monomer H hydrogen bond with the 5th monomer. What happens to the 3 intermediate monomers? So in this case, they're in a coil state. So they are essentially free, technically free, to rotate however they wish to in between 1 and 5. But in a way, if these 3 monomers are free to rotate, they're actually not quite comfortable because 1 and 5 are already locked into place. So 2, 3, and 4 really don't have the freedom that they're supposed to have. So we would call monomers 2, 3, and 4 energetically unfavorable because they want to rotate, but they can't. So we now have to ask the question, what is the next favorable conformation? So monomer 1 is going to start the helix transition by creating a hydrogen bond with number 5. Because 2 is uncomfortable, it's going to want to create another hydrogen bond with the next adjacent monomer, which is number 6. So if we had the 6th monomer, then 2 would have hydrogen bonded with 6 due to energetics. And once we do that, we take a look here. Here, our second hydrogen bond is formed between monomers 2 and 6. So all of these blue ones are the monomers, and we now have to take a look at 3 and 4. So 3 and 4 are supposed to have freedom to rotate, but now they're even more rigid than before. They're even more energetically unfavorable. So you are going to want to form a 3rd and a 4th hydrogen bond. Apart from that, I want to say that forming the extra second hydrogen bond actually doesn't require the ordering of any extra monomers because they're already in place. Therefore, it makes sense that the first hydrogen bond that is right here, this one is going to be the hardest one to form, and then the rest of them kind of just fall into place because it's what they want to do. So after that, we know that there's going to be the first hydrogen bond between 1 and 5, then between 2 and 6, 3 and 7, and then 4 and 8 to create an alpha helical structure. So this essentially is called a cascade effect. Once you have that first monomer start that hydrogen bond to number 5, then all of the remaining monomers in between will be in an unfavorable position and will want to form hydrogen bonds with another monomer after the first pair of hydrogen bonds. So to write this in a sequence, you would have 1, 2, 3, 4 starting the hydrogen bond. So you'd have H, 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 and then a bunch of C's after. Here is another representation of the sequence with hydrogen bonds, where each colored line is a hydrogen bond in this 1D sequence. So we can kind of see this cascade effect going up. So now would be a good time to actually go through a couple of examples of what would or would not be a favorable sequence. So in this course, it's highly likely that we would assign you one of these sequences, and you would have to do some analysis to prove whether it's likely or unlikely. So in this example, it's asking, is the following sequence highly possible or impossible? Explain why. So looking at the diagram on the left, we know there's 2 hydrogen bonds, 1 to 5 and 4 to 8. So we can represent this on the 1D sequence as a connected H bond between 1, 5 and 2, 8. So I've gone ahead and done that. So we take the first monomer in that 1D sequence that is H bonded as H, and we're going to take the fourth one as the first one that's H bonded. So we have H, C, C, H, and then a bunch of C's afterwards. Now, is this favorable? We already know that if there's a rigid structure and there are monomers in between, then it's not going to be quite favorable because these ones want to rotate freely and they can't. So the answer is no. Looking at the sequence, we have 4 monomers that are not hydrogen bonded and want to freely rotate but cannot. So it is energetically unfavorable, and therefore this sequence is highly improbable. So what we would want to say is that in this case, there are 4 extra ordered monomers, which is not favorable at all. Now let's take a look at a second example. Looking at the diagram, we now have 3 hydrogen bonds, 1, 5, 2, 6, and 4, 8. So is this sequence likely? We first draw our lines to say 1 is hydrogen bonded to 5, 2 to 6, and 4 to 8. Once again, 3 and 7 are supposed to be free monomers and free to rotate, but they're in such a rigid form that they cannot do so. So therefore, this conformation is highly improbable because there are now 2 extra ordered monomers causing an energetically unfavorable event to take place. So this sequence is also impossible. Now we want to take a look at the full helix. What would the sequence be? From our examples before, we had 4 monomers in one ring going from bottom to top. We had the bottom 4 monomers being hydrogen bonded to the ones on the top. So this would mean every monomer is H bonded for a full helix, except at the top of the helix. Because at the top of the helix, when your protein sequence runs out and you don't have any more amino acids, you will not be able to hydrogen bond with anything else. So at the very top of that helix, you're going to have the 4 remaining non-hydrogen bonded components in a coil state. So therefore, the sequence is going to be H's all the way up until the last 4, which would be CC, CC. So you're probably thinking, why did we choose to have monomer 1 bind with monomer 5? The reason for this is actually because each turn of the alpha helix has 3.6 residues. So using an I+4 spacing allows us to add 3 more atoms to the hydrogen bonded loop compared to a tighter helix, and therefore it becomes a better model for us to study from. So there is a picture that we've presented before, this one. So these are the hydrogen bonds in the alpha helix. We call this J, and we have J+4 over here because one alpha helical turn is 3.6 residues. So it's much better to model it every 4 instead of every 3. So we always go with J+4. So why do we even care about this? So we do care because we want to be able to figure out how many helix states there are compared to coil states in a solution. And because of that, we care about DNA and protein denaturation. So temperature usually causes transitions to take place between helix to coil, coil to helix, and these transitions are called cooperative. Cooperative meaning the amount of helix changes in a sigmoidal way as a function of an external variable like temperature, and the sigmoidal curve is going to steepen with increasing size of the system. So being able to calculate the cooperativity or the ratio of helix to coils in a solution will allow us to determine how much denaturation or unfolding of DNA is taking place from helix to coil. So when we talk about a cooperative trend, it essentially means a sigmoidal curve like this. These are different curves for different N-mers. So we can see that the longer the polypeptide chain over here, so basically different N-mers, the longer the polypeptide chain, the steeper and more pronounced the sigmoidal curve. What does this mean, and what can we get from this graph? It means the larger the polymer chain, the more sensitive it is to temperature changes. The shorter the polymer change, the less sensitive it is to temperature changes. The theta represents fractional helicity, and that is something we're going to be talking about next before we can fully interpret this graph. So this graph is just here for now. Note that this curve represents a longer polypeptide chain, and that's it for now. We're going to talk about this theta value in the next section and then come back to this graph later to explain what it means in detail. So now it's time to put everything we've learned together through some mathematical models. Essentially, these are models that govern what we call phase transitions, essentially from a coil to a helix. Small changes in temperature or a solvent can cause a polymer molecule to undergo a helix-coil transition, so that it causes two strands of DNA to zip together or cause a protein or RNA molecule to fold, or vice versa. So we're going to introduce a term called subunit cooperativity, which can be positive or negative. Basically, what that means is whether it's easier or more difficult for something to happen involving multiple units. So if we have positive cooperativity among adjacent DNA molecules, then it'll make sure that it's easier to unwind the DNA as a whole group of adjacent nucleotides. So positive cooperativity among adjacent amino acids would also mean hydrogen bonding will allow for an easier helix to coil transition or vice versa. So we're going to start with some of the models that we can use and then move towards being able to fit our data into several different mathematical models. So we're going to start off with something called the Lifson-Roig model, which you've seen before. It's the one we just used in the previous section. We're basically going to assign each residue in a polypeptide either a helical H or a coil C residue state. And this means that we're looking at a 1D sequence model. So essentially those blue circles that we had before, where we had monomer 1, 2, 3, 4, 5, they would be labeled each as H or C, and that would be the Lifson-Roig model. It means we can assume that the total number of possible states in an N-mer, whether it's 5 or 10, a polymer with N residues, the total number of possible states would be 2 to the power of n, because for each of the N-mers, there are 2 options. So it would be 2 to the power of n for the total number of molecular states possible for helix or coil states. So if we know that each state or each monomer can have 2 different states because of the 2-state system of the Lifson-Roig model, what happens if we can choose the statistical weight for each one? So that means we can start applying some math and probability towards our model. So if we can assign individual residues a statistical weight, it means that the product of these weights will give rise to an overall statistical weight for a specific configuration of a polymer of N monomers, or an N-mer as we call it. So then it will tell us whether something's highly probable or not probable. So what kind of statistical weights can we assign using the Lifson-Roig or LR model? We're going to define 3 different residue weights. There is a reason for these 3 weights. We're going to define U as the residue weight assigned to coil states. So we will just call a C state a coil, which is a coil state, and assign it a statistical weight of U. But then what happens if you have neighboring monomers? So we call V the residue weight that's assigned to helix states with a neighboring coil state. Then we have W, which is the residue weight assigned to a helix state without a coil neighbor. So therefore, we have 3 different options between adjacent monomers. We have CC for coil states. We have CH, which gives us the V residue weight, and then we have HH, which gives us W. So the reason for this is because when we were taking a look at whether there were favorable or unfavorable interactions, we noticed that if there was a hydrogen bond before the coil state, then most likely that coil state will also turn into a helix state. So the weight itself is going to be significantly higher than if there were no helix state that came before. So we want to make sure that the statistical weights are based on local interactions that govern the chances of a certain state taking place. If you have a coil state before, then most likely what comes after a coil state is a coil state. If you have a helix state that comes before, most likely it's going to be another helix state. So there's going to be areas where helix and coil states beside each other will have lower statistical weight because it's rare for that to happen. So what is the weighted average of the property over all states? We would have to use a summation to add up all the probabilities. We say that the weighted average A is the sum of each state's probability, right here, times its weighted value. So where do we go from there? We now introduce something called the partition function Z, which describes the statistical properties of a system in thermodynamic equilibrium. It's a dimensionless pure value. And for our purposes, we just want to focus on the canonical partition function. So what this means is that the system is allowed to exchange heat with the environment, but at a fixed temperature, volume, and a certain number of particles. And the definition of the canonical partition function is given by this summation here, where Z is equal to the summation from 1 to n for the number of monomers you have, time summation of that of e to the power of negative f over kBt. And so that term over here seems very familiar. We've seen it before. We know that S entropy is equal to kb ln of W, where W is multiplicity. So we can actually rewrite everything and solve for W to get W equals to e to the power of S over kb. And if we want to introduce temperature into everything and rewrite entropy in another equation, then we have e to the power of F minus E over negative kBt, which we can simplify to negative F over e to the power of kBt. So we've seen this before. We're just now trying to put everything together so that we can use the partition function to figure out the statistical properties of the system we have. So key thing, the takeaway message from here is we have an equation for the partition function, and we have made the assumption that every weight is equal to its multiplicity W. So we want to make sure that the multiplicity ties in with our model in some way. So now we're trying to put things together. Remember the 3 residues we had before, U, V, and W. What happens if we simplify it to 2 residues instead? So just U for a coil state and V for a helix state. Since the weight of every single microstate that we have is equal to its multiplicity, we can essentially say that U is equal to WC and V is equal to WH. So that already gives us some sort of statistical weight or multiplicity for each of those states. So WC, which is the multiplicity of the coil formation, would be governed by bond angle, phi and psi conformations, and WH, which would be the multiplicity for the helix state. These would be governed by hydrogen bonds, and with that, there's going to be a hydrogen bond free energy, which we call epsilon. So just know where those terms comes from and why we use these types of terminologies to describe the statistical weight for the two different states we have. So the next thing, we have to divide all states by the weight of C. Why is that? If we can divide all the states by the weight of C, then what this is going to do is give us a reference point for further analysis and comparison. So we're basically trying to normalize and simplify things. So now that means if we divide the weight of C by the weight of C, we have WC equals 1. So for every single coil microstate, the weight is 1 because it naturally occurs. When we take a look at the weight of a helix state, we have to consider the two options just like before. We have a helix state after another helix state. We give this a weight of S. And then for every helix state after a coil state, we have the weight given as sigma S. So now we have this new value sigma that we have to worry about. And at the end of the day, I know we went through the Lifson-Roig model. We went through the different statistical weights for the 3 microstates we looked at before for U, V, and W. We took a look at 2 different multiplicities for the 2 overall states, helix or coil. And now we're trying to put everything together to try and give some value to each of these microstates within a sequence. So we have to ask ourselves, what is sigma? And is it greater or less than 1? So if we think about it, we looked at a helix coil transition before. The chances of getting a helix state after another helix or a coil state is different. So we have to differentiate the two of them. The chances of getting an H after a coil state is going to be lower than the chances of getting a helix state after a helix state. So based on our analysis from the previous section, remember, it's always harder to get that first helix state transition. So it's always harder to turn a coil into a helix for the first time. Afterwards, it's a cascade effect. So therefore, it's expected that sigma is a very, very small value that's lower than 1, so that there's a lower statistical weight given for a microstate like this. So overall, this does make sense because the first hydrogen bond will always be the least favorable and the least probable to take place. There has to be a large input of energy in order for that first thing to happen. So therefore, sigma is less than 1 and is usually in between 10 to the negative 4 and 10 to the negative 2. So that's very, very low. We're now going to take a look at a dimer as an example. So a dimer is essentially a polymer of N equals 2. So we basically have 2 monomers. So we can obtain a list of all possible sequences of a dimer by combining them. So we have CC, CH, HC, HH. Those are the only possibilities when you just have 2 monomers. You can have 2 coil states side by side. And with these 2 coil states side by side, we know that their weight is 1 times 1. So that's for each of them. And then for CH, we know that C is always going to be 1. We know that H that comes after a C is sigma S. One of the things to note is because you do have a dimer where one monomer is stuck to another monomer, I mean, in this case, we're just going to assume sigma is equal to 1. We cannot make that assumption as soon as we move up to a longer polypeptide chain. So we're just simplifying this for now and making sure sigma is equal to 1 so we can do the math. Now H and C, we have H being S on a standalone basis, and then C always being equal to 1. HH would just be S times S. So the weights are basically the product of individual weights. We can make it easier as well. Instead of writing all this out, you can use binomial expansion to quickly get all of the possible sequences. And apart from that, it's also important to note that the probability of the entire sequence is proportional to the weight of the entire sequence. So if we can calculate the entire weight of the sequence using this, then we'll be able to figure out the probability of that sequence as well. So let's go through an example together of a dimer. In an upcoming assignment, you'll have to take a look at a trimer case or even a longer polypeptide chain where the math does get a little bit longer. So in these cases, it's very good to use computational techniques so that you're not doing everything by hand. So in this course, because computational physics is not a requirement, we are just going to do everything by hand just so that things are a little bit easier. So we're going to take a look at the probability of each of the sequences we looked at before. This is the probability of 0 H's, this is the probability of 1 H, and this is the probability of 2 H's. So we can simply divide the multiplicity of the sequence by the total multiplicity for all of them. So for all of them, just remember sigma was equal to 1, so there should be no sigma in this equation. If you're doing a trimer, you should include the sigma. So the denominator here is always long, so all of the different multiplicities added together would be 1 times S, S times 1, S times S, and 1 times 1. So these are all the different possibilities, and the weight of CC is simply 1 times 1. So this would be the probability of getting your CC sequence. Now for the probability of getting 2 helix states, or probability of getting 1 helix state, you have the weight or the multiplicity of CH plus the multiplicity of HC divided by the total, and you get this. And of course, the last one, it will simply be S times S divided by the total. So when we're presenting these solutions, obviously we want to simplify. We don't want to leave everything expanded in such a format. So because the denominator is quite cumbersome, we want to try and simplify it a bit. And that's where the partition function comes in handy. So we can introduce it here. If you calculate Z2, you actually get 1 plus S to the power of 2. So we can simplify the denominator by just writing Z2. So that's something simple that we can use moving forward. Now is a really good time to define fractional helicity. So we introduced this before in terms of knowing how many helical and coil states are in a solution so that we know how much has been denatured, so on and so forth. So fractional helicity can now officially be defined as the ratio of helix states to the total number of residues or monomers in the sequence. Keep in mind, it has to be the number of helix states because that is the title, fractional helicity. So we're going to go through an example to try and find the fractional helicity of a dimer. So second example, we're looking at the same thing in a different way. We want to find the fractional helicity of a dimer. So we have to first calculate the number of H's we have. We know there are 0 H's for the CC case. We know there is going to be 1 H for the 1H cases, and we know there's going to be 2 H's in the HH case. So those are the total number of helix states multiplied by the probability of getting a helix or getting that state for each one. When you combine everything together, you end up with 2s over s plus 1, like this. And the fractional helicity is then going to be the number of H's given by this divided by the number of monomers, which is 2, and you get S over S plus 1 as the fractional helicity of our dimer. So how important is this value? Let's try to see how it changes as we increase the number of monomers we have. So taking a look at the N-mer case would actually be quite beneficial. So you can use binomial expansion to find all of the possible sequences. And let's say that you get this. Let's say you want to find the multiplicity of a single helical state out of all of your N states. You can represent it like this, where we want the multiplicity of 1H given N states, or N monomers. So C plus H to the power of N will give you all your possible sequences, and then this will give you your weight. Step 2, we want to find the partition function. That is simply going to be 1 plus S to the power of N. So you can try to work that out yourself. So in step 3, you get a general term for the probability of N number of helical states, which is then going to be this divided by your partition function. So just remember, it was the multiplicity divided by the partition function. And then afterwards, to find the fractional helicity, we had to find the average number of H's with all cases. So in this example, we have the summation of the number of H's we're looking for multiplied by the probability of the number of H's in a certain sequence. And then we divide all of those by Zn. So looking at this, I'll go through this more in the live lecture. As we continue, we can simplify everything and get ns over s plus 1. So in this case, we end up with ns over s plus 1, and the next step to find fractional helicity is to divide the number of H's by the number of monomers. So we're dividing by n. We end up with the exact same equation as the one for dimer, and that's basically the take-home message. When we're taking a look at fractional helicity, it's one equation. No matter how many monomers you're looking at, for all polymers of n number of size n, the fractional helicity is always S over S plus 1. So the most important thing when it comes to taking a look and trying to figure out these values is to figure out the statistical weight of getting a helix state. So everything seems to work out very nicely. So for a sigma equals 1, the fractional helicity is given as theta equals to S over 1 plus S. And just remember, we did make the assumption that sigma is equal to 1. So the parameter S then gets a new name. It's known as the relative weight for H, because that is the most important component that we have to find in order to figure out the fractional helicity. So S before was defined as the weight of H divided by the weight of C. So we've defined this already, and we know that it's going to be the probability of a helical state divided by the probability of a coil state. From previous slides, we know that the probability of a helical state is given by e to the power of the hydrogen bond free energy divided by kBt, and the bottom would be the multiplicity of a coil state. So being able to define S like this allows us to go back and relate this to temperature, because we now have a temperature term in here. So with the kBt term, we can now go back to the graph that we had before and do a little bit more of an analysis. So first of all, we have to ask ourselves whether a given polypeptide is going to assume a random coil or an alpha helical structure. So that depends on the competition between conformational entropy and hydrogen bond formation. Might be confusing at first, but all this means is that we have to take a look at the polypeptide's composition, its thermal environment, and its chemical environment, and see whether the polypeptide itself is going to naturally prefer a certain state, or if its spurred on by the formation of hydrogen bonds. So we can test this type of transition using polarized light to get optical rotation values, where we can see a total conversion of a sample from one state to the other state upon a temperature change of just a few degrees. So this is one way of testing how your polymer can change from a coil to helix state. So now we want to take a look at a similar graph to what we looked at previously. So before we had some sort of grayish diagram that had a similar sigmoidal curve. And I mentioned that the longer the polypeptide chain, or the larger the N value, the steeper the curve. So I want to share where that information came from. And that came from Zimm and Bragg's data fitting model, where they used different temperatures and took a look at the optical rotation of monomers. So if the optical rotation is in the negative, you have a random coil state. If the optical rotation is positive, it's going towards an alpha helical state. So basically, if we take a look at this, we have 4 different chains here. We have a very long polypeptide chain with 1500 monomers. We have a shorter one with 46, then 26, and then a fully random coil. So a random coil, it makes sense that it's always got an optical rotation at the bottom of the y-axis. And for a long polypeptide chain, it's going to have a much steeper curve, and it reaches the alpha helical state a lot faster. But what's most important is figuring out what the trend is for N and where we can tie in that sigma value. So as N increases, we know that there is going to be a steeper, sharper curve based on optical rotation. So it means that with a larger polypeptide chain, it's going to be more sensitive to changes in temperature. So a slight change in temperature is going to cause a greater transition from coil to helix state. Whereas if you have a shorter polypeptide, a larger temperature will be required for that transition to occur, and will take a lot longer as well. It may not even reach it at depending on what temperature you have. So apart from that, we have to figure out how the sigma factors in. Remember, sigma was the probability of getting a helical state after a coil state. And we know that it's very hard to do so for the first H monomer. So the chances of getting an H after a coil state are almost 0. So the transition becomes sharper as well after this. So overall, sharper curves are going to come from larger temperatures and smaller sigma factors. So now that we know how we get a graph with the sigmoidal curve from optical rotation, and we know how to model the helix or the coil to helix transition, we can now go back to the original diagram that we had, which was based off of fractional helicity and some sort of temperature. So I'm quickly going to go forward. This is the graph that I presented before with a cooperative trend. I definitely want to go through this again, but we note that it has a similar curve, but the temperature scale is different, and our y-axis is now fractional helicity. So that's why we had to go back and talk about fractional helicity first. So previously, Zimm and Bragg discovered that for this particular solution, the value of S was equal to e to the power of 0.00614 times T minus Tc, where Tc is the critical temperature and Tm is the midpoint temperature. So essentially, we can now rewrite the equation for fractional helicity to be, because it's S over 1 plus S, we can have theta equals to e to the power of this over 1 plus e to the power of this, which gives us that sigmoidal curve from before, like so. So looking back at our graph from before, we can now see that fractional helicity is related to temperature relative to some sort of midpoint, Tm, for long chains from optical rotation data. So pretty much the same graph, just a change of variables. So continuing on, looking at Zimm-Bragg's experiments, we noticed that we had to increase the temperature relative to some midpoint in order to increase the number of helical structures. Big question is why? It's because each act of hydrogen bonding is going to release solvent molecules that are previously bound to the chain. So we know that the change is in the Helmholtz free energy given by delta F is equal to E minus Ts. But in terms of the entropy, we have entropy from the conformational change and entropy change from the solvent. So if we assume that the change in bond energy is greater than 0, then that means at a higher temperature, the free energy is going to become more and more negative. So as the free energy decreases, we know that the hydrogen bonding becomes more and more spontaneous. So that's just based off of our analysis from before. We can conclude using Helmholtz free energy equation and the assumption that delta E is greater than 0, then any decrease in free energy is going to result in spontaneous hydrogen bonding. Okay, so if anybody noticed, we previously said sigma should be really small, but before we said sigma was equal to 1, so that our calculations could be more simplified. Using that simplification, what would be the total weight of a trimer? So let's try to solve this together. First of all, for a trimer, you have all these possible sequences, which you could get from just by writing out all the options or the binomial distribution. You have all the weights that are written out, and this time we want to introduce sigma. To find the probability of each conformation, we need the partition function Z3 this time, which is given by this, and you can work it out yourself as well, or just write it out yourself. You can do the calculations for the partition function and end up with this solution in green, but there is a better way to calculate Zn, especially as n gets larger. You definitely do not want to do this by hand if you have like Z20, or like in the graph, Z equals, or Z of 1500. That would be a nightmare to solve. So we can assign an appropriate weight to a sequence and then use something called matrix formalism to find Zn. So keep in mind, this is not required for this course, but it's here in case you choose to use it in the future. So we'll go through what matrix formalism is for a trimer. The Zimm-Bragg model takes the cooperativity of each segment into consideration when calculating fractional helicity. So you can write the full partition function for a chain of n residues using these matrices. You have C and H in the first one to represent the statistical weight vector, and then over here, which would be like 1 and then sigma s like this. Then you have the next 2x2 matrix, which is going to represent the monomers' conditional statistical weights. So there are 4 possible combinations of the CH together. So we had CC, CH, HC, and HH. So this will give us all the possible combinations. And then we multiply by a column vector of 1s to sum over the statistical weights of all the sequences. And if you do this, you get the answer in a lot less work. And of course, if you could use a computer to solve for this, even better. It'll be faster. So let's now take a look at hydrogen bonds in water. Any molecule that can form hydrogen bonds can alternatively compete to form hydrogen bonds with water. So what this means is that there's competition. It means that hydrogen bonds formed between 2 molecules that are dissolved in water are relatively weak because there's always competition for hydrogen bonding. So to wrap up this section, I wanted to present something that makes a helix not form its usual shape, called a helix breaker. So we're going to introduce proline. Proline is an amino acid that's known as the breaker of helices. The reason for this is because it helps to stabilize a hydrogen bond between the first and third monomers versus the first and fifth monomers. So if you have proline, it's going to stabilize a hydrogen bond between a different set of monomers and thus destroys or changes your entire alpha helical shape completely. So this actually causes the helical structure to break and start to misfold, and proline is commonly known as something that promotes protein misfolding. So that's just a nice summary as to what happens when things go wrong. So as a quick summary of what we learned today, there's still a lot more to go through. We learned about the two-state model of alpha helix to coil transition, or Coil-Helix transitions. Definitely know about the visual representation of the model, so that would be the blue circles. You should understand the difference between impossible versus possible sequences based off of that type of model. Then we took a look at the statistical weights of Coil-Helix transitions. So definitely be able to use the Lifson-Roig model to represent these transitions. Know about the fractional helicity using the weight of coil-helix states, and know about the configurations determined in various ways. So I say matrix formalism here. You don't have to use matrix formalism. Just be able to find a way to figure out all the configurations of a polypeptide chain, whether it's dipeptide or a trimer, or n equals 4 or 5. So being able to write it out and be able to assign statistical weights to the 3 microstates, helix, helix, and then coil, helix, and then coil, coil, those would be the most important skills to know moving forward.